Antigen presentation is a process of the immune system to capture, process, and present antigens to the T lymphocytes. The antigen-presenting cells involved include dendritic cells, B lymphocytes, and macrophages. In this animation, we will show how phagocytosis contributes to both antigen presentation and cytokine production. When pathogens, such as microorganisms, enter the body, phagocytes, which include macrophages, are attracted to the area of invasion by chemotaxis. After attachment to specific phagocyte surface receptors, the pathogen is engulfed into the cytoplasm within a phagosome. This phagosome fuses with lysosome vesicles containing digestive enzymes to form a combined vesicle known as a phagolysosome. Notice that the pathogens are then digested inside the phagolysosome. Let's now take a closer look at the exogenous pathway of antigen processing and presentation. Major histocompatibility class II molecules are produced in the endoplasmic reticulum of an antigen-presenting cell. As the MHC class II molecule is formed, it's transported in a vesicle to a phagolysosome which contains digested particles or peptides of the engulfed pathogen. In addition to alpha and beta chains of MHC class II molecules, a third chain called the invariant chain is synthesized as well. The invariant chain blocks the peptide binding groove of the MHC class II molecule. During the fusion process of these two vesicles, invariant chains are degraded, digested, and peptides take their place on the MCH class II groove. The MHC class II antigen complex is then transported to the cell surface, where it is now accessible for interaction with CD4 T cells. Now, let's focus our attention on the interaction of an antigen-presenting cell with a CD4 T cell. There are several steps involved in T cell activation. This process is initiated by the binding of naive T cell receptors to the MHC class II antigen complex of an antigen-presenting cell. This allows for the antigenic specific nature of T cell activation. Let's take a closer look. Co-stimulatory molecules, which include B7 and integrin located on the antigen-presenting cell, bind to the T cell via CD28 and IgCAM, respectively. MHC class II molecules on the macrophage bind to T cell receptors, or TCR, on the T helper cell. This is followed by CD4 binding to the MHC class II molecule. Next, B7 binds to CD28. Integrin then binds to IgCAM on the T cell. The binding of these co-stimulatory molecules provides a second signal. A third signal results from cytokines, such as interleukin-1, interleukin-6, and tumor necrosis factor, being secreted by the antigen-presenting cell into the T helper cell. These cytokines induce the proliferation and differentiation of T cells into effector cells and memory cells. Activated T cells then produce and secrete interleukin-2, which assists in the development and differentiation of CD4 T helper 1 or CD4 T helper 2 cells. T helper 1 cells produce interferon gamma. When interferon gamma interacts with macrophages, this increases their phagocytic ability and their capacity to digest pathogens. Now, let's proceed with the interaction of the CD4 T helper 2 cell with B cells. CD4 T helper 2 cells only activate those B cells that recognize the same antigen as they do. When a B cell presents a specific antigen recognized by a T helper 2 cell, the bond between the two cells is strengthened. The B cell becomes cross-linked to the T helper 2 cell. T cell receptor binds to the MHC class II peptide conjugate, which contains the antigen. Next, CD4 on the T cell binds to MHC class II on the B cell. This complex synthesizes CD40 ligand, which interacts with CD40 on the B cell surface. The final signal delivered by the T helper 2 cell is the release of cytokines into the B cell, which include interleukin 4, interleukin 5, interleukin 6, and interleukin 13. This induces the differentiation of B cells into antibody-secreting cells and memory cells. Notice the turn of events. The B cell becomes activated, followed by proliferation. And differentiation. Creation of memory cells. And then production of antibodies. Now that we have seen the illustration of the exogenous pathway, let's proceed to the endogenous pathway of antigen processing and presentation. Proteins synthesized by endogenous antigens such as viruses in the cell cytosol are routinely degraded in proteasomes. The endogenous peptides from these proteins enter the endoplasmic reticulum through a peptide transporter known as the TAP complex. In the endoplasmic reticulum, they have the opportunity to bind to freshly synthesized MHC class I molecules. The MHC class I protein complex is then transported to the cell membrane, where it can now be presented to CD8 T lymphocytes. Interaction of an antigen-presenting cell with a CD8 T cell. The interaction between a T cell receptor and the MHC class I antigen complex on the surface of the target cell is recognized by cytotoxic CD8 T lymphocytes. CD8 then binds to the complex, which consists of TCR and MHC class I antigen. This leads to an infusion of toxic granules into targeted cells. Perforin creates pores in the membrane of the target cell, which allows granzymes to enter. Granzymes later activate Cas spaces, which in turn trigger processes that lead to cell death.